Review first, guys. We've defined osmosis 3 different ways. Jackie, tell me one way we defined osmosis. Tell me a little bit more. High concentration of water to a low concentration of water. Jonathan, tell me a second definition.
The movement of water from a low solute concentration to a high solute concentration.
Right. And Catherine, can you tell me the third? Yep, so 3 different ways. Well, guess what? Today we're going to get a 4th way to define osmosis. And this 4th way is important for cells with a wall. Because what other variable other than just solute concentration is important if there's a wall? Pressure. Okay? Today we're going to talk about pressure. And we're going to talk about a concept called water potential. And you're going to see this equation a lot the next few days. First of all, if you paid attention to your handout, what is the name for that symbol right there? Psi. What's psi? PSI. It's a Greek letter psi. But what I want you to think about, it looks an awful lot like what? The trident, which was whose sphere? Poseidon, who is the god of? Water, the ocean, the sea. So water potential, that's why they chose it. Water potential takes into account 2 things. So far, when we talked about osmosis, we've only talked about this one. What's the S stand for? Solute. And as long as you're talking about cells without a wall, that's probably the only one that's really important. But that wall allows a buildup of pressure to happen. This stands for pressure potential. This is the pressure potential. This is called solute potential. Solute potential plus pressure potential equals the total water potential. Now, a couple of key things you need to know about water potential. Number 1, pure water has a water potential of what? This was in the reading too. Pure water in an open container has how much water potential? 0. So that's a water potential of pure water. 0. But here's the important part of this to understand, and the part that can be confusing if you don't pay attention. Just a normal cell, how much pressure is it going to have inside? A normal cell, just been soaking in a normal isotonic solution. How much pressure is it going to have? Not much. Essentially 0. Let's just leave it at 0 right now. Now cells always have solutes in them, right? Sugars, salts. The cell can't live without some solutes inside. It turns out that more solutes mean what happened to this number right here. The more solutes there are inside that cell, what happens to this number?
Increase.
It becomes a bigger number, but it becomes a bigger negative number. So, important thing to understand, more solutes means that this number, this solute potential, becomes a bigger negative number. Becomes more negative. All right, one other thing I'll mention here, and then I'll put up an example. Let me give you that 4th definition of osmosis. Osmosis is the movement of water from high water potential to low water potential. Movement of water from high water potential to low water potential. That's our 4th definition of osmosis. And remember, this idea of water potential is important for cells with what? With a wall. So I'm gonna get a blank slide up here so I can show you a picture.
High water potential
to low water potential. High water potential to low water potential. I'm going to put in a blank slide. All right, so let's say Let's put our equation up here first, just so we got that on our mind. And let's say we had a cell, plant cell. That's just been soaking right now in an isotonic solution.
I'm sorry, how many juniors do you have?
We've got a cell, plant cell, with a wall. It's been soaking in an isotonic solution. So right now, what's the pressure inside that cell?
0.
Essentially zero, not much. There's not a whole lot going on in there. We're going to put that cell into a big cup of water. What's the water potential inside the big cup of water? What's the water potential of a cup of water? Zero. So water potential out here. Water potential out there is 0. Now remember, this cell has solutes inside it, sugars, salts, different stuff. What's the solute potential like inside that cell?
Negative.
It's a negative number. Okay? Probably a pretty big negative number because plants have a lot of solutes, right? A lot of sugars, that kind of thing. So I'm just going to make up a number for this solute potential right now. I'm going to say it's -4. Realize that's just a made-up number for right now to make my example. Question?
-4.
-4 is the solute potential. 0 is the pressure potential. So right now, my total water potential inside my inside my cell is -4. Inside my cup, it's 0. So what's water going to do?
It's going to move out the cell to the...
What's the definition of osmosis in terms of water potential? Higher water potential to lower water potential. 0 is bigger than -4, right? So water's going to go in. And as more water goes in, water starts to build up. What happens to this pressure number?
It's going to...
It starts to increase. There's turgor pressure begins to build up, right? Let's say for right now, maybe that goes up to +1. What's going to happen to this solute number as more water goes into that cell? As water goes in, what happens to the solute concentration? It goes down, which is going to make this become a what kind of negative number? A less negative number. Higher solute concentrations mean the more negative that number becomes. So as water goes in, our solution gets diluted in here, and this number becomes less negative. Let's say for right now, for our purposes, let's say it goes to -3. So that means now, inside our cell, our water potential is -2. Is water going to continue to move? Water potential in here is -2. Out here it's still 0, right? Water continues to go in. Let's say, for sake of argument, that this now goes up to 2. The pressure goes up to 2. And let's say, for sake of argument, that this drops down to -2. So what's our water potential now inside our cell? 0. Water potential outside is 0. Water potential inside is 0. What's just stopped? Key point. Osmosis didn't stop. What kind of osmosis stopped? Net. This water's still moving. It's just if some moves in, about the same amount moves out, right? But here's a really key point. Is this cell at equilibrium? Is the cell at equilibrium? No. Not even close. Because where's all the solutes? In the cell. They can't get out, right? So why did net osmosis stop? What made it stop? The pressure. Okay? More water still wants to go into this cell. If water could want, it wants to go in. But it can't because what's pushing it out? The turgor pressure. That's why cell walls are important. Cell walls are going to stop osmosis from happening when enough pressure inside that cell builds up. Would that ever happen with an animal cell? It just keeps swelling and swelling and swelling until eventually it bursts. So quick review again of what we're saying. First of all, water moves from higher water potential to low water potential. Water potential is made up of two components: the solute potential and the pressure potential. The more water that's in a cell, the higher the pressure is going to be. The more solutes that are in a cell, the more negative the solute potential is going to be. The water potential of pure water is always zero. So if there's a lot of solutes in a cell, the water is always going to go which way? If you put a cell in a cup of water, and that cell has a lot of solutes inside it, which water's going to go in. Okay? Until what keeps it from going in anymore? The pressure. That's the key thing. All right, let me go to a little different slide here. Talk about one other idea. Let's talk a little bit about what's called endocytosis. Literally, what does that word mean, endocytosis?
The taking in of large particles.
Yep. Taking things inside a cell, right? Endo means inside a cell. First caveat here. Endocytosis is another form of active transport because it involves what? Using ATP. Stuff has to be moved. But there's two kinds of endocytosis, two main kinds. Phagocytosis, which we talked about before, and pinocytosis, which we're going to talk about today. Literally, what does the word phagocytosis mean? Cell eating. And this is a good definition right here for phagocytosis. Process of engulfing large, and I want you to add the word solid right in here. Process of engulfing large solid particles and enclosing them within invaginations of the cell membrane. Large solid particles. That's phagocytosis. And after that thing has been engulfed, what's going to get turned loose on that thing that got engulfed? What's your cell going to send to that vacuole? The lysosomes and the enzymes from those lysosomes. And they're going to perform what process on whatever got engulfed?
Intracellular digestion.
Intracellular digestion. Which means that food is going to be digested where? Inside the cell. Intracellular digestion. Carried out by the lysosomes. We're going to see all that stuff happening in the real picture here in the real video. But before we do, we back up one more slide. Let's go ahead and define pinocytosis. Anyone have an idea what that word might actually mean? Pinocytosis. Not so much small. Dissolved substances. The word literally means cell drinking. Cell drinking. And we're going to use this same basic definition. We're going to say it's the process of engulfing large amounts of dissolved solutes. Process of engulfing large amounts of dissolved solutes and enclosing them within invaginations of the cell membrane. So we're only going to change, we're going to take out the word particle, and we're going to say process of engulfing large amounts of dissolved solutes and enclosing those within invaginations of the cell membrane. So it's the cell drinking. Now, very often, receptors are involved in this process. Receptors are made out of what? Proteins. And what will happen in a lot of cases, the solute, whichever solute we're trying to take in here, will bind to receptors, and that will trigger the cell to pull those things in. So sometimes pinocytosis is called this. Receptor-mediated endocytosis, because it's a receptor, a solute binding to a receptor that triggers it to happen. So pinocytosis is sometimes called receptor-mediated endocytosis because a receptor triggers it to happen. Now this pinocytosis is the way a lot of hormones will get into your cells. Certain enzymes can get in and out of cells this way. And also a lot of materials from your blood can travel into and out of your cells through pinocytosis, cell drinking. Why do I have HIV and other pathogens written down here? Why is that on the pinocytosis slide? Exactly. HIV is like one of those door-to-door salesmen, sort of. They come around and they knock on your door. Sometimes you let them in, right? It's probably not a good idea to do that. But HIV knocks on the door of what cells? What kind of cells does HIV affect? There's a specific kind of white blood cell. Anybody know what kind? They're called the helper T cells. And we'll talk about them more later, but helper T cells. And it turns out if these cells get wiped out, which is what HIV ends up doing to them, your immune system doesn't work. But the way that HIV gets into the helper T cells to begin with is it binds to a receptor for some other chemical. And when it does, what does the helper T cell do? When HIV binds to that receptor, it pulls it inside. So like it knocked on the door and it invited it in. And then once it got invited in, it slaughters the helper T cell. Okay? Not a good thing. There are other viruses, other germs that have learned how to trick your cell into pulling them in. They use receptors for other things. And once they bind to those receptors, your cells pull them in, unknowingly pulling in something that's really, really dangerous. All right, I'm going to turn this off, and I'm going to show you a video of some actual endocytosis.